Hello everyone, I'm Yoji Sato from Japan National Institute of Health Sciences. First of all, I'd like to thank Bernie to invite me again to this exciting meeting. Today, I'd like to talk about recent progress in induced pluripotent stem cell-based therapies in Japan. This is a disclaimer. I'd like to begin with current status of clinical applications of iPSC-derived products in Japan, and then I will introduce R&D support for iPSC-derived products by Japan Agency for Medical Research and Development, AMED for short. The last topic is regulatory science on emerging safety issues for iPSC-derived products. Now let me get started with the first topic. This slide shows you clinical studies of iPSC or ESC-derived products in Japan. As you can see, many types of products have been successfully applied in clinical studies with no serious adverse event reported so far. The rows in pink indicate no commercial clinical researches under the Act on the Safety of Regenerative Medicine, RM Safety Act for short. The green rows indicate commercial clinical trials under the Pharmaceuticals and Medical Devices Act, PMD Act for short. So they are based on different legislations. This slide shows you regulations for regenerative medicine and cell therapy in Japan and the United States. As you can see, there is a remarkable difference between the two countries. In Japan, commercial distributions and clinical trials of cell therapy products are regulated by PMD Act, whereas cell therapies under discretion of medical practitioners and non-commercial researches on cell therapies are regulated by the RM Safety Act. In contrast, a common regulation applies to both in the United States. The PMD Act regulates the development and clinical trials of cell therapy products intended for marketing authorization and the distribution of cell therapy products that have been approved for marketing. The RM Safety Act regulates regenerative medicine and cell therapy, which are performed in hospitals and clinics using processed cells that have not been approved for marketing. Non-commercial clinical researches on regenerative medicine and cell therapy using processed cells that are not intended for marketing authorization is also regulated by the RM Safety Act. So in Japan, medical practices using unapproved processed cells are not illegal under the RM Safety Act. This may be like the hospital exemption in the EU. Of course, the RM Safety Act requires a review for such a treatment by a government-certified institutional committee, as well as a notification of the provision plan to the regulatory authority. The RM Safety Act has contributed to protection of the public health since its implementation. As you can see in this slide, in order to prevent future adverse events, the government can arrest medical practitioners who conduct cell therapy without notifying the authorities. As I told in the previous slide, non-commercial clinical researches using processed cells are also within the scope of the RM Safety Act. This table summarizes the differences in regulations between cell therapies provided as medical practices or non-commercial clinical researches under the RM Safety Act and cell therapy products distributed under the PMD Act. Please note that GCP compliance is mandatory for the ethical procedures, but not mandatory for the data system in non-commercial clinical researches under the RM Safety Act. Also, please note that the research plans are not reviewed by the regulatory authorities, but by government-certified institutional committees. As you may have already known, the PMD Act has a special approval system, which is called Conditional and Time-Limited Approval System for cell therapy products and gene therapy products. Under this system, cellular and gene therapy products with confirmed safety and presumed efficacy can receive marketing authorization with conditions and a time limit, including post-marketing surveillance and informed consent for patients. Well, where is the clinical researches under the RM Safety Act? The answer is here. So why this position? In general, it is difficult to fix product specifications and manufacturing process of cell therapy products prior to the first in-human clinical trial. It is also difficult to change the specification and manufacturing process after the initiation of clinical trials because it is difficult to demonstrate the comparability of the product quality before and after the change. Non-commercial clinical researches on cell therapy products under the RM Safety Act are reviewed by government-certified institutional committees rather than by regulatory authorities and have low quality assurance requirements for clinical data. Therefore, this system enables low-cost and rapid initiation of first-in-human studies and provides a flexible approach to optimize the quality specifications and manufacturing process based on the very early clinical findings before initiating commercial clinical trials under the PMD Act. Let's move on to the second topic. This slide shows the support programs for the research and development of cell therapy products in Japan. As you can see, they cover everything from basic research to commercialization. In particular, the steps from basic researches to early clinical trials are supported by a funding agency called AMED, which consolidates the budgets of relevant ministries. AMED was established in 2015. It's not so long ago, but it has promoted medical research and development from basic research to clinical applications by funding to implement the politics of relevant ministries for scientific research and development, public health and pharmaceutical affairs, and industrial promotion. AMED has also contributed to fostering the environment for medical R&D. For example, AMED has formed a network among the major research centers to develop regenerative medicine and cell therapy using iPS cells. CIRA, the Center for iPS Cell Research and Application at Kyoto University, serves as the hub of this network. CIRA at Kyoto University has streamlined R&D at each research center by procuring raw materials for iPS cells and establishing iPS cells in batches and distributing them to the research centers in Japan. As a result, as shown in the blue cells in this table, iPS cells derived from Kyoto University have been used as the raw materials for a lot of iPS cell-derived products that have been developed in Japan. So, the Japanese government is promoting the R&Ds and clinical applications of iPS cell-derived products with a view to their commercial distributions by implementing policies of the relevant ministries for scientific research and development, public health and pharmaceutical affairs, and industrial promotion on a common platform, namely AMED, and by having allogeneic iPS cells as raw materials shared among developers in Japan. The last topic is regulatory science on emerging safety issues for iPS cell-derived products. You can find the term regulatory science in the previous slide. So what is regulatory science? According to the definition of US FDA, regulatory science is the science of developing new tools, standards, and approaches to assess the safety, efficacy, quality, and performance of all FDA-regulated products. So why is regulatory science necessary for the development of advanced therapeutic products? It is because the development of evaluation methods often do not catch up with the rapid development of new types of products, such as cell therapy products and gene therapy products, which emerge as a result of technological advances. It is also because even when new types of analytical tools, such as next-generation sequencers, are developed as a result of technological advances, their capabilities and limitations when used to evaluate the quality and safety of therapeutic products are unknown. From this slide, I will talk about tumorigenicity of pluripotent stem cell-derived products. Tumorigenicity is one of the major concerns for pluripotent stem cell-derived therapeutic products because cells transformed during the manufacturing process and residual undifferentiated pluripotent stem cells may form tumors in patients. However, until about 20 years ago, no characterized test methods existed for evaluating the tumorigenic potential of cell therapy products. So, we have developed several in vitro and in vivo test methods for detection of transformed cells in cell therapy products. Let me show you an example of the method development. Soft agar colony formation assay is an in vitro assay to detect malignant transformed cells. Let me briefly explain the principle of this assay. Adherent normal cells cannot survive when dispersed to single cells. On the other hand, malignant transformed cells or cancer cells can survive in such conditions. Therefore, when adherent normal cells are dispersed to single cells and cultured in soft agar, where they cannot migrate, the normal cells die, but the cancer cells survive and form colonies. This method has been used for more than half a century in chemical mutagenicity assessment and anti-cancer drug development. It is a well-known method, but the problem was that nobody knew its limit of detection. We found this method detect cancer cells only if they were present at a ratio of at least 1 in 1,000 normal cells. This sensitivity is not sufficient at all to detect a trace amount of cancer cells in a cell therapy product. So we tried increasing the sensitivity. Digital software colony formation assay is an image-based analysis method using a high-content image cytometer. Partitioning a cell sample into multiple wells of culture plates enables digital readout of the presence of colony in each well and increases the sensitivity for their detection. In addition, dual fluorescence staining of formed colonies and dissolution of soft agar enable to accurate detection of transformed cells with the imaging cytometer. And digital soft agar colony formation assay has achieved the ability to detect cancer cells in normal cells at a ratio of 1 in 10 million, which is comparable to the ability to find one zombie in New York City. We have also developed in vitro and in vivo test methods to detect residual pluripotent stem cells in cell therapy products. Let me show you another example. Highly efficient culture assay detects residual undifferentiated pluripotent stem cells in cell therapy products using highly efficient culture system, which favors the growth of pluripotent stem cells. This assay can directly detect a trace amount of undifferentiated pluripotent stem cells by measuring the number of colonies originated from a single pluripotent stem cell. This method is quite sensitive. The detection limit is as low as 0.001% and still has a possibility to become more sensitive by improvement of culture system or colony detection method. As a result of improvements, we recently achieved the ability to detect a single iPS cell in 5 million differentiated cells, which is comparable to the ability to find one alien in Los Angeles. So, the regulatory science research has contributed to clinical applications of pluripotent stem cell-derived products in Japan through providing test methods for the assessment of their tumorigenicity or safety. Those test methods have been consolidated as a guideline document and shared among stakeholders in Japan. In addition, we have conducted multi-site studies for validation and improvement of the test method in collaboration with several pharmaceutical and healthcare companies, as well as some CROs. We also keep discussing the quality and safety assessment of cell therapy products on some domestic and global platforms. For example, we have been communicating with members of HESI-CT tracks, which stands for the Committee of Cell Therapy, Circulation, Tracking, and Safety of the Health and Environmental Sciences Institute. We recently published a position paper on challenges and the need for global consensus in the tumor genetic assessment of cell therapy products. HESI-CT tracks consist of 38 organizations and more than 120 participants from industry, academia, and regulatory bodies. CT Tracks has two working groups, namely, one is part of Administration and Biodistribution Working Group, and another one is Tumor Genetic Working Group. And the Tumor Genetic Working Group is also making efforts for validation and improvement of the in vitro test method for detection of tumorigenic cells in cell therapy products. So, in Japan, regulatory science research is being conducted under the policy of promoting the development of new therapeutic products using iPS cells, and various test methods have been developed for new safety issues such as tumorigenicity. Through validation by domestic and international collaborative efforts of stakeholders, these test methods are expected to be utilized as a means to efficiently deliver new iPS cell-derived products to patients around the world who are waiting for a cure. That's all from my side. Thank you for your attention.